Hi, welcome to this week's lab on microscopy, where we will be also incorporating the concept of osmosis. There are two important techniques that I'll need to demonstrate for this practicum. The first one is creating a wet mount. The second one is safely operating a microscope. So let's start with a wet mount. So first thing, grab your slide, and it's good standard practice to always label your slide. So grab a permanent marker. We are creating slides with samples which have been immersed in different concentrations of salt. So the first one that we're creating is a 0.5. You'll also be creating a 1, a 3 and a 5 tablespoon concentration sample as well. Okay? So we now have our labelled slide. The next thing that we need to do is grab our sample. We'll be using red onion. Now, the easiest way to grab a sample of this is to grab a segment of onion. You've got your dull side and your shiny side. What I'd like you to do is to snap the onion towards the shiny side, and as it's just hanging there, pulling it off to the side, you'll have a thin membrane. So grab that thin membrane as carefully as you can. You don't need a lot, but grab some and place that onto your slide. So now we have our sample on the slide. Now, you don't really need to stain a red onion sample, but we're going to stain it anyway because there's some important techniques I need to demonstrate. Firstly, what you'll need to do is to douse your sample in the salt solution. So a couple of drops building up a bubble of water just like that. So that's just to make sure that it's at the correct concentration. Okay? What I'd like you to do then is to dry your sample gently, and then go ahead and add your stain. So you'll need a couple of drops of stain. This is quite a dark stain. Make sure that the stain is in contact with the entire sample. Okay? Just like that. So now we need to apply the cover slip. So gently grab the cover slip, and using a sharp object, place the cover slip on its side and then gently lower onto your sample. Okay? Now at the moment the sample is completely unreadable because there's too much stain. The way to fix this is that if you grab some tissue, place it on one end of your cover slip, capillary action will draw water across. What we can do is tidy that up even more. If you apply water to the other side of the cover slip, that capillary action will draw over any excess water and it will give you a nice clear sample. So once you're satisfied you've washed your sample, go ahead and dry your slide completely. And there is your wet mount sample. Great. Now that you've got your 4 samples, let's go over to the microscope to actually have a look at our samples. So before we begin, just a quick orientation around health and safety and parts in terms of structure and function of the microscope. Now, the proper way to carry a microscope is one hand on the neck, one hand underneath the base. So if your students are transporting the microscopes around the room, make sure that they've always got 2 hands on the microscope and definitely no running. What we have here is a monocular light microscope. So we're using light to actually view our samples. Monocular because there's only one eyepiece. You can also have binocular, but monocular is just fine for our purposes. So the part of the microscope that we look into, and where I've got my camera attached right now, is known as the eyepiece lens. That sometimes will have magnification on it. Generally that is 10%. Now that is on a turret which revolves around, so if you're working with a partner you can actually move the turret around rather than moving the whole microscope itself. Okay? Also, as part of that turret is the nose piece. On that nose piece, you've got your objective lenses. Now, the code for these lenses, there is a color code, but in terms of size, that's another way that you can tell the magnification. So small lens, small magnification, large lens, large magnification. Because this is a light microscope, we also need a light source. So underneath you can see the light source. We have our stage, that's where we put our sample. To focus our sample, we have the gross adjusting knob, which moves the stage a large distance. We also have the fine adjusting knob, which moves the stage a small distance. And we use that for focusing at the end. Now, the proper way to use a microscope is obviously to turn it on and to switch magnification objective lens to the lowest magnification, and that's the smallest lens. The next step is to move the stage all the way to the top of the limit of where it can move. Place your sample underneath the microscope, and if you've got clips, then go ahead and use those clips to secure your sample. When you are now going to focus your sample, you only ever move the stage down. When you're focusing, don't move the stage up, because that can lead to damaging the lens. Now, the reason why we're using a camera is because it's going to be so much easier when you are demonstrating how to focus for your students. So switching now to the camera view, what you can see on the screen now is blurry vision of the sample, and that's what it's going to look like when you first have a look at your sample. So with the stage all the way up at the top, what you are to do now is either using the gross adjusting knob or the fine adjusting knob, now focus your sample. And what's really good about using the video microscope is the students can actually see when it's actually coming into focus. So there is a good focus point. I'm just going to move the sample around to something a little bit more interesting. Now, a couple of other things to note is if your sample is far too bright, a MacGyver hack for you is to just grab some tissue, so either tissue or paper towel, and if you place that over, so just watch how the image changes. Okay? You get more contrast and you can actually see your sample better. Okay? Now, one of the real benefits of using an app like this is that there are no more microscope drawings. So all you do now is you press the camera icon and it takes a photo for you. If you are looking at living organisms underneath your sample, you can actually record. There are a whole range of other functions within the app. So you can draw lines to draw, you can draw shapes, you can write and label. There's some text there. You can also do measurements as well, okay? So there's a whole range of benefits to actually using an app like this. But let's come back to our sample. So now that we've taken a photo with our sample on low resolution, let's go to the next size objective lens, and we're going to refocus now. Now, notice how the image is blurry again. So all we do now, we don't touch the gross adjusting knob, we only now use the fine adjusting knob, and we can focus down till we actually get to the samples, and here we go. So there you can see on the screen the sample at medium magnification. Now I'm going to leave it to you to, I'll take a photo here. I'm going to now leave it to you to now switch to high magnification, and when you're focusing on the highest magnification, do not ever touch the gross adjusting knob, and if you're using the fine adjusting knob, only ever focus down. So if you do lose focus, what I do recommend is that you go back to medium, refocus, then come back to the high. Okay? So go ahead, take photos at the highest magnification of your 4 samples and go ahead and insert them into your lab book. Thank you.